Good afternoon, and welcome to today's webinar, Does Carcinogenesis Start in the Womb? Endocrine Disruptors and Breast Cancer. My name is Michelle Dixon, and I am the Program and Development Director for the Massachusetts Breast Cancer Coalition, and I would like to thank you for joining us today. I am very pleased to welcome today's speaker, Dr. Anna Soto. Dr. Soto is a professor in the Department of Immunology at Tufts University School of Medicine. She co-authored the book, The Society of Cells, Cancer and Control of Cell Proliferation. Dr. Soto's research focuses on fetal origins of adult disease, stromal epithelial interactions on organogenesis and carcinogenesis, biomechanics of morphogenesis, and theoretical biology. Dr. Soto, welcome.
Thank you very much, Michelle. I want to thank you and Cheryl Osimo for organizing this webinar and for all the work that you are doing at Mass Breast Cancer Coalition. So, and thank you for the introduction. We are going to talk about a theme that I think is very important, and is that breast cancer starts in our very early life before we are born. And this is that has to do with the environment. And so we have to talk about how the environment gets to have such an important role, because it's not only us producing cancer in some instances, but also because it's a very important determinant of our health as well. So I'll start with a brief introduction to endocrinology, because we are talking about hormones. And so therefore we have to refresh a little bit our memory on what those hormones and the endocrine system does. So the endocrine system is a group of cells and organs that produce chemicals that we call hormones. These hormones travel through the bloodstream in order to reach these target cells, the cells that have receptors that recognize these specific hormones. And as you see here, we have a set of organs that produce these hormones and that are very important in many respects. Those hormones are recognized by receptors. A cell that doesn't have receptors for that hormone will be totally blind to the hormone. Here we are looking at what we call nuclear receptors, and in our case, because we are interested in ovarian hormones, for example, we are dealing with what we call nuclear receptors. So the interactions between the hormone and the receptor involve a network of cofactors that go from the binding of the receptor to the hormone to the receptor to affecting some cofactors and DNA ultimately, but sometimes it doesn't happen. It just bypasses DNA, and it's something that it's very important as well, but people forget, because since the 50s, our society is very, very, very much fixed into DNA, to the point that now metaphors about it, like a brand of cars say safety is in our DNA, like if a car will have a DNA. So that is a very dangerous thing. I mean, this is an aside, but it has to do also with what we are going to discuss today, that we are exaggerating the importance of genes in every biological phenomenon, and that should go, because it's not healthy. So what does the endocrine system do? There's at least three types of functions that are relevant. One is to maintain the constancy of the extracellular fluids, so that our cells are bathed with the appropriate conditions for their performance, that we call homeostasis. And this is important through life. They regulate the nutrient supply to all cells of the body. In other words, we eat a few times during the day, but our cells are bathed in nutrients throughout the day. So there is a mechanism there that facilitates this nutrition. And the other function is to regulate development, growth, and reproduction. And those are the hormones that are important to us today because we are going to talk about what happens in the womb. So in order to get into the subject, we have to talk about two different types of effects of hormones. One is called activational effect, and those effects are reversible upon removal of the hormone. For example, during each cycle, we have a process whereby there is growth of the lining of the uterus, and then during menstruation is shed, and then a new cycle starts, and this cycle is controlled by estrogens and progesterone. Okay, so those activational effects are reversible. Then we have organizational effects that are for the most part irreversible. So we are going to talk about the effects of thyroid hormone on fetal and newborn brain development as an example. What it means is that during fetal life, organs are being formed, and you have to have the right complement of hormones, not too little, not too much, in order for things to develop normally and efficiently. So here we have a subject. This is from a very old book of endocrinology, 22 years of age, 3 feet of height. So this person looks like an infant, but it's 22 years old. What is going on there? Well, the distribution of fat in the body and the proportions of the body are not the ones of an adult. This happens because there is decreased levels of thyroid hormone in that situation, mental retardation, increased fat production, and this can be corrected by thyroid treatment early in life, that is immediately after birth. For example, that is why the kids, as they are born, the thyroid TSH is measured in order to know whether or not they have a deficit of this hormone. So this is an irreversible effect. Either you act immediately after birth or you lose the ability to fix this problem. And those are the effects that we think are relevant to this idea that cancer may start in the womb. Okay, and if the deficit of thyroid hormone occurs in adult life, yes, there are characteristics like dry hair and this type of edema that we perceive very well in the tongue and dryness of the skin, etc. But when we treat this patient with thyroid hormone, all this reverts to normal. So what we care about is these organizational effects. But before I get into that, I will tell you a short personal history of how I got engaged with this. And this is the environment, because the environment got into my lab. So in 1989, while we were studying estrogen control of cell proliferation, Dr. Sonnenschein and I at Tufts University were doing our controls were always the same, our positive control and our negative control. And then all of a sudden, we got that our controls that shouldn't proliferated as much as the cells that were exposed to estrogens. So where was this estrogenic activity coming from? And after 4 months of work, we found out that they came from the plastic tubes where we store components of the tissue culture medium. So we found that plastic shed an estrogen, nonylphenol, which is used as an antioxidant, and that also is used to make detergents and spermicides. So nonylphenol was the first estrogen identified in plastic. So this is the environment getting to us. And this made us to work on endocrine disruptors that I'm going to define a little later. So this discovery made us invited to the Windspread conference and to the genesis of the screen assay. That is an assay that we developed that allowed us to discover about 20 chemicals that were estrogenic and are present in the environment. Some of them are pesticides, some of them are plasticizers, etc. And in 1991, the term endocrine disruptors, that is exactly 30 years ago, or it will be in July this year, the term endocrine disruptors was coined at the Windspread Conference. The laboratory accident was used as a basis to elaborate the screen assay that, as I said, allow us to discover that many chemicals used in the environment, be them pesticides, plasticizers, and a lot of other things, are present in the environment. And this was recognized by the Massachusetts Breast Cancer Coalition that presented us with the Marla Phrasing Award in 1999. And so we are still working and fighting the epidemic of breast cancer, as you know. And there is still a lot to do because we are not being helped by an appropriate public health policy. So what is an endocrine disruptor? Endocrine disruptors are exogenous chemicals or mixtures of chemicals that interfere with any aspect of hormone action. And so this is a very important thing, any aspect of hormone action, because as I mentioned, there are many hormone actions, and then they can be always interfering with very important sensitive processes of the organism. So, oh, sorry, I don't know why this happened. So where do we find those estrogen receptors, those estrogen endocrine disruptors? Everywhere. So you see here, you will find them in food because of food contact materials. You are going to find them in detergents, cosmetics, lawn care products, packaging, etc. And Silent Spring Institute did a very interesting experiment about the canned food and the presence of bisphenol A, and how if you eat healthier than that, you can significantly decrease the amount of at least bisphenol A and some estrogens, xenoestrogens in your system. But what interests us the most is the fragile fetus. And why I'm saying fragile? Well, we start being just one cell, right? A fertilized egg, a zygote that divides. And so here we have 4 cells, then we are 8, 16, etc. But then at a given moment, we start a process that is called organogenesis. And so organogenesis is the formation of all our organs. So we become a human here, a baby. And this process is extremely sensitive to hormones. That, as I said, you should have the right amount, neither more nor less. And our knowledge of how bad it could be was given by a misguided treatment. The ethyl-stilbestrol, a synthetic estrogen, was given to women during pregnancy, and this happened here in Massachusetts, actually. The idea was that during pregnancy, estrogens and progesterone are being produced, and that therefore, if you give more estrogens, you would be able to block the danger of miscarriage. Well, this resulted in many, many women, millions of them, being exposed to the ES between 1948 and 71 to prevent miscarriage. But at the same time, experiments were made, epidemiological studies that show that absolutely it doesn't prevent miscarriage. But in 1971, a few women, young women, appear at Mass General Hospital one by one, showing a very, very rare cancer that is called clear cell carcinoma of the vagina. And because they had this very rare disease that appears very infrequently in women that are over 60, they were able to trace this anomaly to exposure of their mothers during the time they were developing in utero to DS. And that had specifically, that exposure leads to clear cell carcinoma of the vagina only if exposure occurs before the 13th week of pregnancy. So this allow the medical profession to describe the fetal xenoestrogen syndrome that appear in girls that were exposed during fetal life to diethylstilbestrol, this synthetic estrogen. So it was observed in women, in humans. As I said, it was given to prevent miscarriage. It happened before 13 weeks of gestation, and the frequency was 1 in 1,000 about. Now, the cancer manifested after puberty, so that means that you need an estrogen in order to make this cancer manifest. So it's like a two-hit. Additional anomalies were found, like malformations of the oviduct and uterus, decreased reproductive success. So these women have problems to maintain pregnancies, for example. And then 25 years later, it was discovered that those girls have an increased risk of breast cancer at the age of prevalence, that is about 50 years of age, and that this effect occurred in women that were exposed in utero, not necessarily before the 13th week, but any time during pregnancy. So that is a very serious side effect. And it was, the FDA didn't forbid the use of DS, but advised not to use it. In any case, this practice practically disappeared in a few years. And that showed us something, that is the environment as the maker of phenotypes. And now we knew that as biologists. I mean, I'm a physician, but biologists knew that since at least 100 years ago, more, 150 years ago, that the environment contributes to making the phenotype. We are what we are because we have our inheritance that comes from our parents, but also because of the environment around us. So it's not your genes that make you or what you are. They are not in the driver's seat. It's a conjunction, a constellation of things, and among them, the environment in which we live. And I give you an example that we knew for many, many years. That is this one. You see, this is a caterpillar. So if it's born during the summer, it looks like a twig. Okay, it mimics the twig because that is what the oak looks like. But in spring, it looks like the flowers. Okay, so this happens in many, I'm not going to dwell on this, but there are thousands of cases in which that happens. In humans, of course, the effects are not that spectacular in the sense of a change, total change of form, but they are very spectacular in the bad sense because of the bad things that do to us. So here we have the idea that the development is a genetic program brought to you by the molecular biology revolution, and in the 1950s or so, that predicated that the fetus is isolated, the mother is just a fetal incubator. And then what we are now thinking that has to do with old embryology, that is what people thought in the 1860s and 70s, but also the new discoveries that happened after this, 1980 or so, this started to change and accumulate evidence that is not the case, that what we have is more than permeability, that the fetus is exposed through the mother to light, to food, to hormones, etc. So the mother is the fetal environment, but this maternal environment is also in communication with the big, wider environment, because things like, for example, those detergents I was talking about, and plasticizers and pesticides get to the fetus through what the mother breathes, eats, and drinks. So we got now to a concept that is fundamental to those of us that work on developmental biology are interested in how the environment determines phenotype. That is what we call a discipline called ecological developmental biology. And our ideas are that that idea of program is demonstrably wrong. There is no central control, and we are not our genes. That's for sure. Genes are just a part of the equation. They do not hold a privileged causal role in what we become. The environment plays a main role on the determination of the phenotype. So there are several participants here, and none of them has the most important role. And there is redundancy and plasticity. And so therefore, the possibility of compensating one part of the determinants with another. Organogenesis is the result of reciprocal interactions among cells. And here we have a classical example that I think illustrates what I want to say. And the idea is that in order to have a kidney, you have to have two tissues here. You don't need to memorize what they are, but it's something that is called the nephrogenic mesenchyme and the Wolffian duct. And if you separate them and put each one of them in a dish, nothing happens. But if you put them together, you obtain a glomerulus, that is the filtration apparatus of the kidney. And how does it happen? Well, there are interactions there. Some of them are movements, some of them are proliferation in one part and not in another. But the interesting thing is that there are many different types of cells that are formed. And those cells are formed by the interaction between these two tissues, that is reciprocal interaction. Although those cells come from the nephrogenic mesenchyme, you cannot get them to be formed from it when this is not put in close contact with the Wolffian duct. So there is a double reciprocal participation. And that is what determines organ formation, be it the kidney or the mammary gland or the breast in humans. So now this brings us to, now we are talking about cancer starting in the uterus. So we have to talk about our cancer theory. That is called the tissue organization field theory of carcinogenesis. And this started because what we observe is that chemicals that are not mutagens, and there are lots of them that are not mutagens but are carcinogens. So how do they produce cancer? And then you have to explain that. And to make a very long story short, because it's more than 30 years old, I'll show that in a diagram what we think. So as I said, in order to form a kidney, you need two tissues, right? Here we have two tissues. This is like a schematic of any so-called epithelial organ, like the mammary gland. So this arrow is a carcinogen. So we know that this tissue, the epithelium, the form, the shape of this tissue is determined by its interaction with the mesenchyme. And if we expose this mesenchyme to a carcinogen, we would alter the reciprocal interactions indicated here with double-headed arrows that exist into these two components of, for example, the mammary gland. So cancer will occur not because of mutation of I don't know what nucleus of what cell, but because you are altering the morphogenesis, that is the formation of that tissue. And this could be just the de novo formation, like in utero, or tissue remodeling, that is what happens through all life. And it happens actually in every menstrual cycle. So when this alteration, with this interruption of these proper reciprocal interactions happens, the tissue starts changing shape, forms a hyperplasia, hypertrophy, etc., dysplasia, and could eventually form a carcinoma. All these processes in this theory are reversible if you know how to stop exposure. So what we are interested in is the tissue level of organization, and more specifically, this interaction between the epithelium and the connective tissue or stroma. The targets of carcinogens are the tissues. Cancer is the problem of tissue organization comparable to organogenesis. And the natural or default state of cells is proliferation and motility, like in bacteria. So the difference is that when they are in a multicellular organism like ours, those cells that spontaneously will proliferate are inhibited from doing so by the structure of the tissue where they reside. This is in a nutshell. I can give you, it will take more than a full seminar to explain the details. But that is in essence what we are talking about. Something that is not a mutagen is affecting the development of the mammary gland. So we now go to the chemical we are studying, that is bisphenol A. And bisphenol A is now the poster boy of endocrine disruption because it's the one that has been more studied. It just does pretty much the same that diethylstilbestrol did in humans. It affects the onset of puberty, alters mammary gland development. Now we are talking about rats and mice. Changes in the uterus and ovary, decrease of fecundity and fertility, so alters reproduction. Changes in brain steroid receptor levels, changes in behavior, hyperactivity, increased aggressiveness, alters sexual behavior, increased susceptibility to drugs of addiction, etc. Predisposition to cancer of the breast and the prostate, and obesity and diabetes, etc., etc. So there is an actual complex syndrome, and that is why the NIHS started organizing meetings with every person that worked with bisphenol A in order to call to the attention of the public health community what was going on, and the Endocrine Society also ran a couple of public statements, one in 2009 and another in 2012, saying that this is a serious problem. And in spite of all this, nothing is happening. But I tell you that in Europe, it is happening now. We'll go to that at the end of the seminar. So what is the evidence that we have of that estrogens are xenoestrogens in the environment could be a factor that induces breast cancer? Well, the hypothesis is that the increased incidence of breast cancer observed during the last half century is due to exposure to endocrine disruptors. You know, the risk for a woman in her entire lifetime used to be 1 in 22 in the times of my mother, and now it's 1 in 7, 1 in 8. Part of it is due, we think, to exposures during fetal life, but also exposures throughout life. And that if there is time, we can talk about that, but I know that you are very much aware that ovarian hormones are a big risk factor. So I will concentrate only on cancer starting in the womb. Epidemiology has shown that gestational exposure to, for example, DDT, increases the likelihood of developing breast cancer in humans. So then experimental studies, like the ones we have done, show that gestational exposure to environmentally relevant, that is very low doses of bisphenol A, increases the risk of developing mammary cancer. So now we will talk a little detour about what is bisphenol A. And bisphenol A is this molecule that is a cousin of the diethylstilbestrol, we say. It has a very close similarity. So it's the building block of polycarbonate plastics, like the ones on your glasses, for example. A component, I mean, the lenses are made of polycarbonate because it's a lot lighter than glass. It's a component of other plastics and epoxy resins like the lining of food cans, and is a prevalent chemical in our daily lives. So BPA is one of the highest volume chemicals produced worldwide. Over 8 billion pounds are produced annually. 100 tons are released into the atmosphere per year. So it's a serious problem. So what did we do in order to understand whether or not the fetus is a target is being exposed? Well, we took, as I said, environmentally relevant doses of bisphenol A and administered that to pregnant animals. And then we look at the mammary gland that was being formed at that moment. So we were looking at embryonic day 18, that is 2 days before birth. And here we see the epithelium of the mammary gland, and that is surrounded by connective tissue. And here we have some islets of tissue that is going to become fat tissue. This blue stuff that you see here is the connective tissue component called collagen, that is a protein of the connective tissue. Okay, this is the control. This is the animal that was exposed to bisphenol A, and you see that the blue stuff is not there. Here we have the epithelium, we have the stroma, but this collagen is organized in a very different way. And then we look at this other protein, here stained in red, that surrounds the epithelium of the mammary gland. It's called tenascin C, very abundant in the control, but is inhibited. The synthesis and deposition of this protein is inhibited by exposure to very low doses of bisphenol A. What does it mean? Because you see, it's a very subtle thing. Well, it means, to just shorten a bunch of other evidence that I'm not showing, an accelerated maturation of the stroma with increased accumulation of fat droplets into these pre-adipocytes. I told you about these islands of tissue that will become adipose tissue. Alter collagen and tenascin deposition that I have shown you already. And as a result of this, what we have is an effect on the development of the epithelial compartment of the mammary gland, that is the organ that the epithelium is what gives the function of the organ. So here we see the animal that is controlled, and here we see the animal that has been exposed to BPA. So we see more development of the epithelium. Now, if that was only advanced growth, it would be like being short or being tall, but it's not the case. At the time that this is accelerated, there are other processes that are being blocked. So what we have is a lack of synchrony between processes that have to occur at the same time. So there is an increased number of these structures. There is an increased area of those tactile systems, but it's delayed lumen formation. This is a solid core that has to form a lumen. That is, it has to become like a tube. And so we see right there that some things are being accelerated, some things are being blocked. So what we did is we started developing an organ culture of the fetal mammary gland in order to see whether BPA produces the same effects, because we wanted to figure out whether the target was the mammary gland itself or was, for example, the other endocrine organs of the fetus. So we exposed only the fetal mammary gland, rudiment, and we see that adding very low doses of bisphenol A, that's exactly what we have observed when we administer it to the mother, that is to increase the development of the epithelial structures. Here is just to show you that this story that chemicals have different effects at different doses. So here at low dose, that is the environmentally relevant dose, we have more growth of the epithelium, as I have shown you before, in the animal exposed in utero. If you give a lot more, 1,000 times more, you see inhibition of the formation of this tactile structure. And this effect is inhibited by anti-estrogens. Okay, so we show here that bisphenol A principal target is the mammary gland itself, although there are other components, but it happens, a big part of it happens by exposure of the mammary gland, the developing mammary gland to BPA. And so now the animal is born and we stop exposing the animal to bisphenol A right after birth. And so what we see here is the growth of the mammary gland goes in this direction. This is a lymph node, and here we have the same structure. So by 1 month of age, we see that the epithelium is invading the stroma, and we see that it has moved far beyond the lymph node. It's growing in this direction. In the animal exposed to bisphenol A, we see a retardation of that growth. So you see, it's interesting, but there is this combination of different events. The animal that has been exposed in utero and is not exposed at this moment to bisphenol A, when the estrogens hit that will make the normal mammary gland invade the stroma, here growth is kind of blocked because of something that happened during fetal life. The other thing we observe is that while the controls have no problem creating a lumen, that is this making out of a solid structure something like a tube with a lumen, we see here cells that are dying. Okay, that is how you get this cavity formed. In animals that were exposed in utero, that is 1 month earlier to bisphenol A, we see that there is no the same amount of cell death. So there is a blocking of the formation of the lumen of the mammary gland duct. If we look at what's happening in the epithelial compartment, here we have a duct, okay, that is this beige-colored structures are the nuclei of cells that are stained with an antibody against progesterone receptor. And we see this cluster of cells here. Those are the points where the mammary gland ducts will branch. And this, right now, we just see that is a premature expression of this natural phenomenon. But if we look at 4 months of age, you see that there is a lot more branching and formation of little structures called alveoli in the animals exposed in utero to BPA. And the appearance of bedded ducts, those ducts that have the structure like you see a pearl string, are intraductal hyperplasias, a precursor of cancer in situ. And so this appears in animals that have been exposed to bisphenol A and not in animals that have not been exposed. So BPA induces ductal hyperplastic lesions and carcinoma in situ. And here we show quantitatively what I showed there. That is that while the controls are okay, have very few hyperplastic ducts and no carcinoma in situ, the animals exposed to all doses of bisphenol A have a threefold increase of these intraductal hyperplasias. And if we look carefully, we start seeing that animals that have been exposed to bisphenol A also have carcinoma in situ. And as you know, women that have carcinoma in situ are recommended to undergo a mastectomy. So, sorry, this is going in the opposite direction that I want. So in rats, we find that not only produce carcinomas in situ, the rat is a better model than the mouse. The other experiments were made in mouse for breast cancer. And so we see here that there are tumors. And when you look at the histology of the tumor, it's the same type of histology that we find in humans. So BPA increased the incidence of palpable mammary tumors in a strain of rats, the ones that we use, that is the SD strain. Now, years after that, I mean, we are talking very recently, the FDA ran an experiment that was a study that is called Clarity, and they found that a significant increase of adenocarcinomas, as well as the combination of adenomas or adenocarcinomas in these animals treated with the lowest dose of bisphenol A in their studies. So FDA is not doesn't like to admit this, and so now I mean myself and the other participants of the Clarity Consortium are battling their interpretation. This is a long story. I can explain some more during the question and answer session. But suffice to say that they confirm what we have shown years ago. How does it happen? Well, here we have our interpretation. So that is a theory of how breast cancer will happen. Part of it has been backed by data that BPA acts in the mammary gland, in the stroma, because there is where the estrogen receptors are. The altered stroma produced delayed lumen formation and accelerated epithelial growth. That is what I show you today. And this is what hasn't been, we do not know the process by which these effects then alter the response of the mammary gland to reproductive hormones. So now the mammary gland responds with twice the effect of than a controlled mammary gland to the same dose of estrogen. Also, the BPA acts in the estrogen receptors of the hypothalamus, altering the hypothalamus pituitary ovarian axis and producing altered ovarian cyclicity and altered secretion of mammotropic hormones. So now we have a double whammy. The mammary gland is affected and responds in a different way to the ovarian hormones, ovarian and pituitary hormones, and the regulation of the secretion of these hormones is already altered. So as a result of all this, mammary cancer appears. What needs more study is what happens here, 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 and there, but the effect is there. And actually, we don't need to know more in order to know. I mean, we want to know more because we are scientists, but we do not need to know more in order to say that this is utterly dangerous. And it shouldn't be happening. It shouldn't be possible that humans are exposed to bisphenol A like this and nothing happens. So, conclusions about these experiments is that the fetal xenoestrogen syndrome challenges current reductionist paradigms and invites an integrative system outlook, bringing biophysics, cell-cell interaction, cell motility, and tissue dynamics to the forefront. So developmental genetics, I mean the study of molecular biology of development is no longer the only game in town. Okay, so we have to be broader and we have to include the environment. Perinatal exposure to BPA triggers a florid syndrome, decreased reproductive success, altered behaviors, obesity, diabetes, and cancer. None of these endpoints are good, and we know that all these pathologies are increasing. So my question is it wise to keep exposing our offspring to these chemicals? Not only to our springs, to ourselves, offspring, to ourselves as well, but we care more about them because the effects are more pronounced when exposures occur in utero. Which brings me to another point that I want to make, that is science for the sake of science and applied science. Now, basic science tends to, people, practitioners like me, we live with uncertainty. There is always a new experiment to be done, a T to cross, an I to dot. So 100 years elapsed between Galileo saying that the the Earth move, etc., around the sun, and the Newton laws of that happened 100 years later. So there is no rush because you don't need this information in order to act. You don't need that to change the orbit of the Earth, say. So what you care about is knowledge by itself, and so you are in no rush. and you can always gain more knowledge. But in medical practice, this is not the idea. I mean, just time is of essence. Physicians have to reach conclusions and act now to prevent or cure disease or save a life. So we are in a totally different predicament. And medical epidemiology has to be acting in a particular way. So if you are testing a pharmacological agent, you choose as default the null hypothesis, that is, no effect expected, because it's best to err on the side of the false negative. In other words, if you want, say, something, a drug that will cure diabetes, you want it to be extremely effective. You don't want to give people something that could have side effects if it's not being extremely effective in taking care of the pathology that trigger your prescription. But if you're a public health epidemiologist, you have to work in the opposite way, because when you are studying exposures to potentially harmful agents, you have to choose the alternative hypothesis. So your default is that a deleterious effect is expected. This is good because it's best to err on the side of the false positive. So it is better to say, well, bisphenol A is bad, and then stop exposure while you study if it's so bad, than say, well, we don't have enough evidence and let exposures happen for an extra 30 years. That is what is happening right now. Since wind spread to today, we have 30 years in which babies and everyone else has been exposed to xenoestrogens, and we continue merrily ignoring it. So which brings me to what I say normally, but here in America, we haven't done too much about it, but enough is really enough. And the Endocrine Society has said that, and a bunch of people, but there is no action at the level of the authorities, that is, those that can regulate exposures and dictate public health policy. But there is a little light at the end of the tunnel. In October 14, 2020, the European Commission published a chemical strategy for sustainability that proposes to establish legally binding hazard identification of endocrine disruptors based on the definition of the WHO, building on criteria already developed for pesticides and biocides, and apply it across all legislation. Ensure that endocrine disruptors are banned in consumer products as soon as they are identified. Now, this is very important, and of course, the industry is already getting armed with their ways to create doubt and produce false information. They are already there. But a great step forward, this great step forward is the result of the efforts of scientists, journalists, politicians, resistance, and scientific societies. Not surprisingly, this event has created a lot of resistance from the usual suspects. And we know that that will soon materialize. As a matter of fact, it happens a couple of times that we saw how they are trying to derail the process. So endocrinologists should be ready to oppose the delaying tactics that are being armed by the industry, with the proper principles and clear understanding of when enough is enough. But to do that effectively, we need the other part of the equation, that is the public. And in Europe, it has been very active in certain countries. So enough is enough has been said. And that, as a result of that, in France, they have already developed, banned the use of bisphenol A in food contact materials and of some phthalates that are also endocrine disruptors. And they have done it before than anyone else on baby bottles. And that is about 10 years ago. And they have said enough is enough with the use of plastics in the canteens, that is in the the dining rooms of colleges, I mean, from kindergarten to university. So this is being happening at this point over there. That is the last, and I testify for all these laws in France. And so I think that here we need the same amount of activism, because it's not only if endocrinologists and physicians try to oppose these delaying tactics. We need the people, through a democratic process, really, to go and knock on the door of their representatives and get things done. So we need to at least be as good as Europe is. And I have to thank NIH for funding my research, since non-Phenol in this area of research. And but 30 years have elapsed, and you would like to see effects here. You would like to see at least what we are seeing happening in Europe right now. So with this, I want to finish my presentation, and this is just a book we published a long time ago about what cancer is, that is being translated into other languages. The last one is in Spanish, and that happened 2 years ago. Okay, thank you for your attention, and I entertain questions, of course.
Thank you, Dr. Soto. Could you speak a little to what happened with the FDA's interpretation of the Clarity study?
Of course I can. This is a very long story, but I have to give you some background. I will tell you. The Clarity study was, at the beginning, the idea was that FDA was going to test bisphenol A using the endpoints that they always use, that are funny things, like weighing tissues, things like that, that where you won't find fine specific effects of the type that we observe when we look at endocrine disruption. And the NIHs, that at that point was, the director was Linda Birnbaum, decided, and this is an idea of Linda Birnbaum and Jerry Heindel, they decided to, or had the idea that this study should open the participation of people that discover these new effects, like myself and Fred Fomsal and others. And so what they did is to open NIH, open a call for applications. So people like myself and Fred and everyone that wanted to apply could apply to try to reproduce our results in the experiment that FDA was running. So the idea was that you would have the old established endpoints that have been always used for regulation, and at the same time, in the same animals or similar animals, the endpoints that we have discovered. The idea being that the authorities like FDA and EPA and all those said that, oh, what happens in academia is not done on what is called good laboratory practice. Good laboratory practice is not a better way to run experiments. It's something imposed in the industry because they used to just tear apart the pages of your protocol books when the results didn't please them, okay? So you could hide all kinds of bad stuff. And then when this regulation was put into action, they couldn't remove data that was against what their chemicals. But they use the excuse, oh, well, people are not scientists in academia, don't do good laboratory practices, so therefore we cannot accept their experiments. It's just a strategy to block regulation. So they say, I mean, NIH said, well, okay, that is what you care about, then you can have the same people using animals that are run by you, which means that they are following the good laboratory practice protocol. They didn't like the idea at all, but they accepted it. And so we had this project. And interestingly, like, for example, they say that those responses have to be always linear and monotonic. Well, not linear necessarily, but monotonic. That is, the more you put in, the more toxic it becomes. And as I told you, this is not the case for endocrine disruptors, but it's not the case for many other things. Just think of yourself trying to salt your food. If you have no salt at all, it's not very tasty. If you put enough for your taste, it will be wonderful. Put 3 times more, you cannot eat it. Same thing happens with vitamins. Like a low dose, you have a problem and you have to, take more vitamin, as people in these latitudes are advised to take vitamin D because we don't synthesize enough. Fine. So if you take 50 times more vitamin D, that will be toxic. So it's not a monotonic dose response. It's not that the more you take, the better it is. It's totally different. And so these other types of curves that are, like I say, if you put a little salt is good, if you put too much is bad, this type of thing is normal in endocrinology. And we have studied these things many, many years ago. So what happened is that when they saw that the results in cancer appear to be significant, very significant at the lower doses than at the higher doses, they decided that there must be an error somewhere. And so therefore, they say that it was caused by, it's a random phenomenon, and which is not. So there has been a lot of reaction. And among those reactions is like, for example, in the study that I did on the mammary gland, my group did, what we did is to show that in the mammary gland, using more refined and much more refined statistics that they are capable to use, and using a new methodology that we developed, in which we could get automatic data because we use software that you don't need humans to intervene. We demonstrated that the dose response curves are non-monotonic. And still they are trying to say that that is bad, the same that they are doing with the other 10 projects. They don't like to see those effects, and they are, well, I don't know what they are, because you have to agree with me that this negation of science could be because they are incompetent, say, because they are disingenuous, all of the above, or because they are fraudulent. Whatever it is, it's not good science. And they have this weight because they are an authority. So I don't know where this is going, but the fact that we had that directive of the European Union that I just described for you is our hope, because it's very well thought. And I think that we have to be supporting that until it arrives here. And it will arrive here like everything else when it's done in Europe. In other words, when in Europe they decide that bisphenol A cannot be in the Nespresso capsules, then you don't have it in the States, because they won't make it on purpose good capsules and bad capsules. Same about cars, emission of gas emission of cars, etc. When the Europeans set standards, then whoever is making that product will accept those rules, because it's stupid to use 3 rules if you can use one, and you use the most stringent one. So Europe is actually improving indirectly our health, if you will, but we have to do more. And I hope that by being very much aware of what happened in Europe, the medical societies could keep an eye in what the industry is doing. But we need aware people, the same way that in Europe people are fighting. And the coalition did a lot in the early days, remember? And we have even a hearing in 1993, etc. But then nothing happened. And I think that we need to reactivate all these sorts of intelligent people, women that care about their health and their children's health, etc., that are well educated and could educate other people. Well educated, that meaning knowing that cancer is something that can be avoided, that the environment has good and bad things, etc. So if we have that type of activism, probably we can help. But if we don't, it won't happen, because we need this massive involvement. And I hope that I can persuade you that we have to continue with the same vigor that we manifested in the early days, in the early 90s.
Absolutely. We do have another question around the FDA. How many emergent contaminants approved by the FDA are endocrine disruptors?
I don't know. I mean, it's like we know that Bisphenol A is an endocrine disruptor. We know that plenty of flame retardants, etc., are endocrine disruptors. And you don't see all the activity that you have to see. But so again, there are people. In order to address this issue, I would give you a name. Maricel Maffini is the person that knows a lot of this, and she has worked trying to get FDA to do what they have to do by law. But I'm not an expert on that. You need special type of people to address each one of these issues. So there are things that can be done there because it seems that unless you sue them, nothing happens, for example. And but when you do, things may happen, and she has been successful together with other organizations. So we can talk about that. I mean, I can provide you with the contact information, but that I think is very important, that we have people that are watching what's going on and trying to use what is already available to improve our health by making these agencies respect what they have to do by law.
Yes. Dr. Maffini was actually with us for our PFAS and food packaging panel discussion last December, and she'll be joining us again later this summer.
Well, let me know when she's talking, because she's so good. And yeah.
Yes, I definitely will.
I
definitely will. Well, Dr. Soto, I have to thank you so very much for your time today and for this discussion. I wish we had more time. There's so many more things to cover. But on behalf of Cheryl Osimo, MBCC's Executive Director, and our entire Board of Directors, I want to thank you very much for the information that you've shared with us today. And I want to thank our many listeners for joining us today. And for those who had logged in and asked questions about whether this webinar is being recorded, it is, and it will be available later today on the MBCC website at mbcc.org. And I wish you all a very good afternoon. Thank you so much.
Thank you. It's a pleasure to be with you all. Take care.
Take care.